Hello, my name is Jacqueline Gunn and I'm a student at Unity, Maine, and I'm going for my wildlife conservation degree. I was hired by the town of Voluntown, Connecticut, to kind of go over a current issue that's occurring in their ponds. So how is atrazine impacting us in our fish? Recently, there has been a devastating amount of dead fish that has washed upon the beach of Hopeful Pond in Voluntown, Connecticut. In order to understand what is going on, I acquired several samples from the aquatic organisms, and I found one thing common, which is a substance in the cells of the liver in every sample. This is the chemical atrazine. Atrazine is a non-naturally occurring chemical that most farmers use in herbicides to kill weeds. Its pure form is white, powdery, and easily dissolves in water, making it hydrophilic. Its molecular structure is C8H14C1N5, and its functional group is 2-chloral, 4-ethylamino, and 6-isopropylamino, and 1,3,5-trizine. On August 15th, 2021, the town of Allentown had received a tremendous amount of rainfall, which led to the runoff of the herbicide atrazine used on farms into the neighboring water of Hopeful Pond. So I wanted to discuss how atrazine is actually being metabolized in the fish. And when we look at the actual anatomy of the fish, this is what we see. So the fish goes through aquatic respiration through a catabolic gas exchange in which it needs to obtain three things: a large surface area to volume ratio, short diffusion distance, and a maintained concentration gradient. Now, looking at the physical anatomy of the fish's gills, you can see that the fish has 4 layers of the gills on both sides of the head. The gills are made up of stacks of gill filaments, and each gill filament is covered in gill lamellae. Positioned at right angles to the filament, this is our large surface area. Right here is our large surface area, okay? So when a fish opens its mouth, the water runs in over the gills and then throughout a hole on the sides of their heads. Inside the gill lamellae is a capillary network. When looking at how the concentration gradient oxygen is maintained, it is directly linked to a countercurrent exchange. This happens when the water flows over the gills in the opposite direction of the flow of blood in the capillaries. It ensures the equilibrium is never met, and it ensures the diffusion gradient is maintained across the entire length of the lamella. And this is how the fish is obtaining its oxygen into its bloodstream. So I also wanted to compare how salt is being dissolved into water as opposed to atrazine, because then you can really understand how this molecule is hydrophilic. So when an ionic compound is added to water, individual ions interact with the polar regions of the water molecules during the dissociation process, disrupting their ionic bonds. Dissociation occurs when atoms or groups of atoms break off from molecules and form ions. Consider the table salt's molecular structure, which is NaCl, or sodium chloride. When NaCl crystals are added to water, the molecules of NaCl dissociate into a positive Na and a negative Cl ions, and spheres of the hydration form around the ions. Then the positively charged sodium ion is surrounded by partially negative charges water molecules, oxygen. The negatively charged chloride ion is surrounded by partially positive charged hydrogen in the water molecule. Since many biomolecules are either polar or charged, water readily dissolves these hydrophilic compounds. So this is Alka-Seltzer, just dissolving into a glass of water, but I thought it was a really great example of how it dissolves. And then this is also how atrazine in water forms a product, which is the chloride plus hydroxyatrazine plus a positive hydrogen. So when the atrazine is dissolved in the water, the fish is taking the chemical in and it's entering through its cells via diffusion through the lipid bilayer of the cell's membrane. And then the cells that are in the fish are eukaryotic. So I'm just going to go quickly over what a eukaryotic cell is. So the cells have, the eukaryotic cell is a membrane-bound nucleus, and it has organelles. And organelles are little organs inside the cell, just like we have organs inside our bodies. These are all the cell's organelles. The nucleus contains the cell's DNA. You've got the cytoplasm. You've got the peroxisomes, which detoxifies any harmful substance. You've got the mitochondria, which produces ATP, and then it stores it. And then you have the Golgi Apparatus. The Golgi Apparatus is a place where lipids and proteins can be stored, tagged, packaged, and distributed. Then with the cell membrane, you have the phospholipid bilayer, which is made up of a phosphate group and two fatty acid chains. Then you have fish cells have something what is called gap junctions. They specialize in the transport of ions, nutrients, and other substances that enables other cells to communicate. The rough, smooth endoplasmic reticulum, this organelle modifies proteins and synthesizes lipids. They also have what is called an extracellular matrix made up of glycoproteins and protein collagens. This is going to be attaching to that phospholipid bilayer. You got lysomes. This is where the cells can get rid of anything that it doesn't need, such as proteins, polysaccharides, lipids, nucleic acid, and even worn out organelles. And then you've got the centrome, which its job is to organize the cell's microtubules. Next, I kind of wanted to bring to attention the cells of the liver. You've got many cells in the liver, and a few of them are the hepatocytes. I'm sorry if I'm pronouncing it wrong, but these cells are the chief functional cells of the liver. Their jobs are to perform a number of metabolic and endocrine and secretory functions, such as producing bile, chemical metabolizing, and storage of glycogen and lipids. Hepatocyte liver cells branch off into other cells like hepatic stellate cells. These are the hepatic stellate cell. So this is what it looks like inside of a hepatocyte cell. On a normal day, where healthy macromolecules such as amino acids, fatty acids, sugars, and such as glucose, nucleotides will be broken down in a catabolic process within the cells of the liver and will produce ATP in the mitochondria. And that will be used for energy, but most of the time ATP energy gets stored for other cells and other job functions. So next we have what it looks like inside of the hepatic stellate cell. And this cell is responsible for maintaining the liver's health, but because this is a normal healthy day of the fish, the liver satellite, it pretty much remains inactive. That's because it just really doesn't have a job right now. It doesn't need to repair any injuries that are harming the liver. It doesn't need to detoxify any toxins, nothing's entering bad into this cell. However, let's say one day the fish had taken in some of the water that contained the chemical atrazine. This chemical is now waking up the cell, and so that it can metabolize this chemical into a more preferable substance. When the cell breaks down a toxin, its waste is what's known as a bile acid. However, when the bile acid accumulates and it can't be shipped out fast enough out of the liver, it creates a reactive oxygen species, ROS. A chemical herbicide like atrazine has the ROS products such as superoxide anion O2, and they're considered free radicals. When the free radicals reach a high quantity, it can in turn react with biological macromolecules, leading to enzyme inactivation, lipid peroxidation, DNA damage, and cell death. Here we have a good example of lipid peroxidation, which is also known as the oxidative stress found in the liver. This happens when the ROS-free radicals steal electrons from the lipids and other healthy cell membranes, resulting in the damage and death of those cells. This isn't atrazine specifically, but I just thought it was a really good example. There are enzymes that can help combat and eliminate ROS. ROS is formed, and then the hepatotic cell tissues. There are antioxidant enzymes called superoxide dismutase, SOD, and then also catalyzed CAT. This protein encoded by SOD gene is bonded to copper and zinc ions, which make it an isozyme. The encoded isozyme SOD1 is a soluble cytoplasmic protein found inside the heptic cell, and this acts as a homodimer, which means that it's going to convert the superoxide radical O2 into a hydrogen peroxide. SOD1 is a super important enzyme, and if this gene of the enzyme were to be deleted, then all of the ROS produced would build up, killing off cells, and the organism would eventually die. Also the CAT, so super dismutase, we'll break it down to this, but it's still pretty harmful. That's when you get another friend helping out, such as catalase, and in this we'll break it down into oxygen and water. So why should we care about the fish's health? I would care about the fish's health because it also kind of like represents the water that we're drinking ourselves. If we're living in nearby this area, it's not just their water that's being affected, it's our own water. We are also kind of like eating the fish that come out of it, or something is eating the fish, and then it's like the ecosystem chain where you see the web of what is eating what, and eventually we're going to be eating the chemical ourselves. So then you kind of think, like, if this is what's happening in fish, then it could be happening to me too. In humans, it's known to be an endocrine disruption. It alters the levels of the key hormones, and it could delay puberty. Atrazine causes a kind of chemical castration amongst frogs, and frogs have known to start becoming, male frogs have been starting to change their own gender and produce eggs. There could be reproductive effects such as miscarriage, reduced male fertility, low birth weight, and then it's also linked to a lot of cancers. This atrazine chemical has also been banned in Germany, Italy, Norway, Sweden, just because its widespread presence in the water. Now, I also wanted to bring up the fact of biodegradation, which could be helpful in getting rid of this toxin in our waters. So it's basically, we have these biodegrading bacteria right here, much like our enzymes in the body. It can work to break down the atrazine in the water into a less harmful substance. And for this example, they actually paired it up with a crystal to enhance the bacteria and just kind of like supercharge it, and that cleans it even more. I propose we perform our own studies with biodegradation of this toxic chemical to find a way where we can implement it and clean our waters for our own health and for the ecosystem's health. Thank you.